also analyzed eosinophils under pathologic conditions. Knocking out ATG5 in the eosinophil lineage resulted in delayed and reduced eosinophil differentiation under both physiologic and pathologic conditions. As a result, we observed accumulation of immature eosinophils in the bone marrow and a significant drop of mature eosinophils in the circulation. Moreover, ATG5 knockout mouse eosinophils and ATG5 low-expressing human eosinophils exhibited enhanced degranulation abilities, suggesting that eosinophil effector functions are negatively influenced by ATG5-dependent autophagy. Taken together, we show that mouse and human eosinophil hematopoiesis and effector functions are regulated by ATG5, which controls the amplitude of overall eosinophil immune responses.
Hello everyone, my name is Avishai and I'm a PhD student from Professor Monit's lab at Tel Aviv University. Today I'm going to present you my project about transcriptomal profiling of eosinophils polarization. First, as we all know, eosinophils are classically related to type 2 diseases, such as parasitic infection and allergies. In recent years, it was found new rules for eosinophils, associated them with type 1 environments, such as combating microbes in colon infections. From other myeloid cells, we know that exposure to polarized environment leads to polarization with unique phenotype. So, we were interested to define this polarization state in eosinophils. Thus, we activated eosinophils with classical type 1 and type 2 stimuli. We found, as can be seen by PCA plot, that this stimulation resulted in significant transcriptomic changes in eosinophils with distinct phenotype. The greatest difference was observed between IL-4 stimulated eosinophils to eosinophils which were stimulated with interferon gamma plus E. coli. Finally, by bioinformatics analysis, we discovered that different stimuli induce distinct pathways. Thank you.
Hi, my name is Josh Mitchell, and I'm a graduate student in Dean Mosher's lab at the University of Wisconsin-Madison. We are interested in determining the differential cellular processes that commit eosinophils to terminal endpoints after activation with the cytokines IL-5 and IL-33. Laura Muehlbauer, a collaborator from Josh Kuhn's lab at UW-Madison, used multiplex tandem mass tagging to compare phosphosites in human eosinophils treated for 10 minutes with IL-5 versus nothing, IL-33 versus nothing, or IL-5 versus IL-33. Each comparison was made on 5 pairs of samples, giving us great power to find differences. Roughly 36,000 sites were identified, with approximately 9,500 significantly changed sites. Upon cytokine activation, a set of common biological processes, cytoskeletal rearrangement, vesicular trafficking, ubiquitination, and splicing, were all upregulated. IL-5 activation resulted in upregulation of JAK-STAT signaling, and IL-33 activation led to upregulation of NF-kappa B signaling. These datasets give us a detailed glimpse into the biological pathways required for general activation of eosinophils, as well as the pathways that commit eosinophils to IL-5 and IL-33 activated states. Thank you.
Hi everyone, my name is Timothy Fetrelet and I'm a PhD student under the supervision of Professor Hans-Uwe Simon and Professor Shida Yosefi at the Institute of Pharmacology at the University of Bern in Switzerland. Today, I would like to share a recent report where we studied the link between eosinophil degranulation and mitochondrial DNA release within the process of eosinophil extracellular trap formation. We investigated the time-dependent release of the granule protein EPX and mtDNA following C5s stimulation of both human and mouse cytokine-primed eosinophils. We observed the maximal EPX release and upregulation of the surrogate marker CD63 already after 1 minute of stimulation, while mtDNA release only reached a maximal level after 30 to 60 minutes. These findings suggest that the molecular pathways leading to degranulation and mtDNA release are at least partially different, and that the association between granule proteins and mtDNA scaffold is likely to occur in the outer space. Thank you for your attention, and I look forward to discussing with you in the breakout room.
Hello, my name is Yash Choksi, and the title of my poster is Eosinophils May Be Protective in the Development of Murine ESCC and Alter the Tumor Microenvironment. In this poster, we demonstrate that eosinophils are present within areas of human ESCC and in a significantly greater number in early stage versus late stage disease. We then show the same is true in a 4-nitroquinolone 1-oxide murine model of ESCC. Next, we determined that modifying eosinophil number, either with an anti-IL-5 monoclonal antibody or in an eotaxin-1 knockout mouse, resulted in significantly worse disease. Next, we showed that in mice treated with an anti-IL-5 monoclonal antibody, there was not only worse disease, but also a significantly greater number of CD4 positive T cells. Finally, we showed that in vitro co-culturing an eosinophil cell line, or EOL1 cells, with cancer cells resulted in a significantly greater apoptosis. I look forward to discussing future directions or any questions any of you might have about my work. Thank you so much.
Today, I'm reporting inflammatory and regulatory eosinophil gene expression in an in vitro model of eosinophilic esophagitis. Eosinophils have pleiotropic functions in tissue environments and may adapt regulatory or inflammatory phenotypes. Outstanding questions in this area include what roles different phenotypes play in inflammation, what factors promote differentiation, and what changes define that differentiation. We examined gene expression by circulating eosinophils and eosinophils co-cultured with esophageal epithelial cells with and without IL-13. Of the published inflammatory eosinophil genes, the majority were upregulated in co-culture and to an even larger extent with IL-13 supplementation. By contrast, regulatory eosinophil genes were by and large detected at the lowest level in IL-13 supplemented co-culture. Evidently, eosinophil gene expression is highly responsive to the local cytokine environment and may adapt an inflammatory phenotype in IL-13 supplemented co-cultures. Future studies will assess whether eosinophil functions such as cytokine production change in these conditions.
Conflicting data has emerged regarding a role for eosinophils in IgA production, with some reports that eosinophils support IgA levels during homeostasis. In my study, where wild-type and eosinophil deficient double-gata mice were purchased from the same vendor and bred as littermate controls, I found that eosinophils are not required to maintain homeostatic levels of secretory IgA. When I went on to measure circulating antibody isotypes in these mice, an interesting phenomenon emerged. When these genotypes were born from separate dams and either co-housed as adults or co-reared their entire life in the same cage, I consistently found a significantly elevated level of circulating antibody isotypes in double-gata mice. But when I repeated these assays in littermate controls, the antibody levels were always the same. Together, this indicates that rearing conditions can impact steady-state antibody phenotypes, and emphasizes the importance of optimally controlling rearing and housing conditions throughout life between mice with different genetic backgrounds.
I think we have completed our 7 abstracts for the breakout room. I thank Nina Jermick, Avishai Dolitsky, Josh Mitchell, Timothy Fretelet, Yash Choksi, Julia Dunn, and Rachel Fitzpatrick for their teasers, 1-minute teasers into their research. It was fascinating. Thank you so much. I'm going to open the floor for discussion. It can be in any phase or format. We have about 10 minutes for discussions. Thank you. Any burning questions? Anybody? Please feel free to unmute yourself and ask the question to the presenters themselves. Anybody? Okay, so I have a question for Julia Dunn. Do I have Julia Dunn on the, yes, I do see her. Julia, you presented, it was a very interesting one. You presented a gene expression profile, and you said there were regulatory genes for eosinophils that were suppressed on IL-13 co-culture. Which specific genes were you talking about? Sorry, it just went in a flash, that's why.
Yes, that is, so it was in the text of the abstract, and I had thought that, but let me pull that up. So yeah, not all of them were downregulated. It was actually really, it's interesting, kind of the mixed phenotype, but it was sort of the same for the inflammatory genes. A lot of them are really high in the co-culture with IL-13, but a lot of them were up at baseline. But in terms of the regulatory genes that were down with the co-culture plus IL-13, some of them were RUNX3, IL-17 receptor. SELL is a big one, and one that we've been looking at the protein level, because it's pretty straightforward to just, it's a surface marker, so that we can We can measure it by flow, and we have been recapitulating that. EMR1, CCR3, or just a couple of the other genes that were that have also been sort of reported to be associated with the regulatory phenotype that we saw as being downregulated in our model. But again, all of that information about, what really defines a regulatory and an inflammatory eosinophil is, it's sparse. I think that these words have been used a lot, but in terms of, and also, kind of what are we looking at in a mouse versus a human eosinophil? So I think a lot of this is up for grabs. And if my real feeling is that there's more than two flavors, and we're really just kind of scratching the surface.
Yes, that might be the scenario. There's been a recent paper that just came out in JCI by Dr. Anuradhare and Dr. Sally Wenzel looking at IL-18 in severe asthma. Did IL-18 come up as one of the genes that you investigated that may be affected by IL-13 environment or the epithelium?
It's not in this particular kind of sub-study, no, but that is something that I had been looking at a lot. It came up as one of the cytokines, actually one of the only cytokines that was specifically upregulated in our co-cultures. I think was actually more likely to be produced by the epithelial cells, just based on the RNA-seq data. The eosinophils were differentially expressing receptor subunits. And so I do think that IL-18 signaling is involved, but I'm not sure about the eosinophils like producing IL-18.
And it was the co-culture with the epithelium, right?
Yes.
Right. Okay, I've hogged enough of the space. Anybody, please? Questions? Any question to Jas Chosky or Josh Mitchell? That was some interesting as well. I think.
Question for Nina, if that's okay.
Yep.
So really interesting kind of teaser that you gave there. I was just wondering why you kind of started looking at ATG5 to begin with, and whether you kind of have a working hypothesis now moving forward for why it might be inhibiting eosinophil effector function, because certainly if it's doing that, it could be, potentially a really interesting therapeutic target for future studies.
Yes, hello. Thank you for the question. So why ATG5? To answer first, it's because the lab works on autophagy and was already looking in the ATG5 previously also in neutrophils. And indeed, it was very interesting to see the differential effects also of ATG5 knockout between neutrophils and eosinophils, seeing that, for example, knockout in neutrophils accelerated and caused accumulation in the different organs, while knockout in eosinophils caused delayed and reduced differentiation. And it was also interesting as well, for example, for neutrophils and mast cells, reduction of ATG5 or ATG7 also reduced effector functions, while in eosinophils, it was also to our surprise that it enhanced, for example, degranulation activity as well as bacterial killing. We don't really have the exact mechanism behind. We also looked for the activation of some pathways, for example, p-STAT3, as well as p38 and p44. We saw that following the priming with, for example, GM-CSF or IL-5 cytokines, we could see increased activity of these pathways when we checked that by Western blot. So we thought that might give a hint for the increased activity of knockout in this case.
Really interesting. Thank you.
Thank you for the question.
Any other questions? How about I found that Josh Mitchell had put I think a small eosinophil figure. I think it's a light microscope image that you had put. It seems that the morphology is different as well with IL-5 and IL-33 trigger. Can you just shed a little light on that? Was it a light microscopy? image?
Yeah, the morphology does change quite dramatically too. So from kind of our peripheral purified eosinophils, they're nice and spherical. And then when you do acute treatment with IL-5, you see a very distinct polarization where the nucleus moves to the back of the cell, kind of forming this nucleopod. And the granules are at this migratory front where they make up this main portion. And it takes on this kind of acorn-like shape. With IL-33, with this acute activation, what you see is ultimately there's kind of almost this initial activation state where, unlike perhaps the more distinct, sharper features of IL-5, it's a little bit more oval shaped. And then eventually, as it becomes more adhesive and more migratory, it actually flattens out into this larger pancake-like cell, which forms this very thin layer, probably about 1 or 2 microns in thickness. And it's this monolayer of granules. And the migration is also much slower than the rest. So that's been one of the inherent interesting questions for us and trying to tie in some of these features. I know it was a 1-minute tease to practically 9,500 phosphosites. So it's a
Great. Okay, no, that makes sense. My next question is to Timothy, unless anybody else has a question.
Oh, sorry, go ahead, Julia. I had a really quick follow-up question for Josh, which I'm just curious to know where you're obtaining your eosinophil culture. Is it differentiated from bone marrow progenitors or?
Oh,
blood?
okay, yeah. So our eosinophils, right? So these are all patient purified eosinophils.
Okay.
But what about the fact that they may already have had the effect of IL-5 or IL-33 in their system, whether they're steroid naive, whether atopic, non-atopic, or healthy?
Yes. So the patients that we're getting our eosinophils from usually have some sort of allergic condition, which just gives us the ability to actually purify them, right? Outside of that, we do have on a patient by patient basis, right, kind of a lot of that health information. Oftentimes, at least from what we found, right, when we get our eosinophils, they are kind of the spherical unactivated state. I can't say I've looked very heavily at each individual state. I will say, at least for the sake of kind of the experiments that we found, is that we've had very good consistency, not only with the morphologies, but also kind of the seemingly activated type between them, right? We always get they're allowed to rest too, which I guess can in some ways help. But we've generally found that from the peripheral blood, we've had very consistent, at least morphological presentations.
Have you looked at least looking at the activation markers like CD63 to make sure that they are unactivated, considering you're getting it from patients?
Yeah, so we do. There's some immunofluorescence we look at. I can't say that we've strictly done flow, looking at all of the surface markers before all these. So that's something that one of the things I do want to look at is take a comparison across kind of this wide variety of patient samples. Take a look at them.
Thank you, Josh. We have about 3 minutes in this breakout room before we have to go back to the main session to wrap up today's webinar. So in the last 3 minutes, if anybody has any questions to any of the speakers or any of the 3-minute abstract speakers.
I have a question for Josh, but it's not actually about his abstract. I've been thinking a lot about eosinophil nuclear shape. And so I guess I was wondering, in any of your kind of studies, so you mentioned about the nucleus kind of getting pushed to one side of the cell in certain activation conditions. I'm wondering, have you seen conditions where the shape, dramatically changes? Maybe like recircularizing or becoming more fragmented, or any kind of phenotypes to that effect?
Oh, that's a great question. So, I mean, in some ways, so I don't see any changes per se in segmentation, right? Usually the 2 lobes do seem pretty consistent across the activation state. I will say, in respect to what cytoskeleton, how that might be putting pressure on the nucleus seems to be kind of interesting, because with IL-5, right, I mean, it really jams in. So I don't have the images up, of course, but in confocal microscopy, right, when you look at DNA staining, really jams into the back. Those 2 lobes are really squeezed together. And IL-33, the nucleus flattens out more, right? I guess it has more room to spread. So at least in that respect, you do see perhaps the compactness or the maintenance of the nucleus into a tighter positioning. That certainly changes between those 2 states.
Interesting. Thank you.
Okay, we have one last question. Anybody? We have to leave the breakout room in about 120 seconds. So any last question for anybody, any of the speakers? I can see Sina Karimi is also joined the call, who had a 3-minute abstract. So any questions to Sina, who just worked on the mepolizumab efficacy of the T-cell driven HES? So if that is it, then I'm going to close the floor for now. Please feel free to leave the breakout room and go into the main session where Dr. Florence Rufos will be taking the session. Thank you.